Hello, my name is Headrick, and I'm here to present on inflammatory and cellular damage markers in a pig model of interventricular hemorrhage with and without MR-guided high-intensity focused ultrasound treatment. Interventricular hemorrhage occurs when blood vessels within the brain rupture and bleed within the ventricles. It's present in 25 to 30% of children born preterm with very low birth weights. The incidence has been decreasing since the 1980s, but the prevalence remains high due to improvements in neonatal intensive care, improving the survival rate of infants born prematurely. Death, neurodevelopmental impairments, and hydrocephalus are all associated with IVH. The intraventricular blood is thought to cause brain injury from localized tissue damage and the toxic effects of prehuman iron on brain development. Inflammation due to the hemorrhage has also been implicated in the pathogenesis of post-hemorrhagic hydrocephalus. Managing the post-hemorrhagic hydrocephalus involves placing a shunt that drains cerebral spinal fluid to limit post-hemorrhagic ventricular dilation to prevent the increase of intracranial pressure. These shunts carry the risk of infection or failure. Thrombolytic drugs have also been investigated, but have not been found to significantly affect neurological outcomes. Other treatments involving blood removal, such as extraventricular drains and neuroendoscopic lavage, have also been tried. However, there's no consensus as to which mode of treatment is best. There's a need for more new therapies for IVH that target the condition specifically to prevent the development of complications, such as hydrocephalus. In the context of thrombolysis, the breaking up of blood clots, MRgHIFU uses pressure to create bubbles from gases trapped within tissue and to oscillate them to generate shock waves to break up the blood clots. To deliver the treatment in patients, we would use a robot adapted from a profound sonolene, which we've been using in our animal studies. Lai et al. Published a study in 2021 using a piglet model of IVH. To briefly summarize, blood was extracted from the pigs and then infused into the ventricle and left to clot. Then some pigs were treated with MRgHIFU and others were not, and the pigs were survived for up to 28 days with samples collected post-mortem. Immunostanding is a process where selected antigens of various markers are detected in samples through staining with antibodies labeled in ways to make them distinguished from other antigens. One of the labels that can be attached to an antibody is horseradish peroxidase, which reacts with diaminobenzodine to give a brown coloration or red, and hematoxylin can be used to stain background tissue blue. Astrocytes are the brain's most common form of glial cells. These cells support the creation and maintenance of neurons and are also critical to maintaining the blood-to-brain barrier, a layer of cells that prevents the brain from being damaged by only allowing in specific molecules from the blood. Glial fibrillary acidic protein, or GFAP, staining can be used to evaluate astrogliosis, which occurs when astrocytes undergo functional or morphological changes. Its upregulation is implicated in many pathologies. CD11B is a marker on non-lymphocytic white blood cells, including macrophages and neutrophils, and on microglia. And it has been used in studies of brain hemorrhage to measure inflammation. Space 3 has been used to identify apoptotic cells. A Zeiss axiozoom light microscope was used to examine the slides. Images were taken along the ventricular walls. The percentage of cells positively stained with these markers was calculated using a QPath. When imported, these images had their typeset as bright fields DAB. Values for DAB staining were automatically generated by typesetting and were adjusted by setting the staining vectors. Annotations were made by outlining the ventricular wall with the brush tool at a 20 micrometer scale. Then the pixel classification tool was used to measure the cells above the threshold in the annotated regions. full resolution and a standard threshold of 0.25 were used for all analyses. These are just some GFAP stained images of the ventricle walls. GFAP staining produces the most visually intense staining when stained with the cortex and along the ventricle walls, but was closest, darkest closest to the ventricles. Analyzing the images, QMath also produced the highest average percent of positively stained cells, but differences between the medians for the two treatment groups were not significant. These slides and the CP11B slides were stained with a variant of DAB that stains red or purple due to a region of some slides being discolored with yellowish brown dots in a way that made it hard to differentiate staining in the region from pre-existing discoloration. Caspase staining was kind of the least intense visually of all the stains and was most intense along the ventricle walls. Differences in the medians for the two treatment groups were not significant. 11B staining was also most intense along the ventricle walls. Analyzing the images in QPAP produced the lowest average percent positive stained cells on these slides, and the differences between the medians were not significant for the treatment groups. So there's no clear effect of treatment on these markers of astromeiosis, apoptosis, and inflammation. Some limitations include slide damage to some of the slides, leading to areas need to be excluded from analysis. Small sample size was only 6 untreated pigs and 4 treated pigs, with the lack of a negative control to act as a baseline. Conclusion: further research is needed on the effects of MRgHIFU on the treatment of IPH. In future studies, it would be best to increase the sample size, add a negative control group, optimize clot lysis treatment parameters, and quantify clot lysis efficacy during treatment. We would like to acknowledge funding from CHR and Hydrocephalus Association, and we would also like to thank our collaborators at Sick Kids Imaging Facility and the Pathology Department at STAR. Thank you for listening to my presentation.